perception that when you talk about a foreign intruder which induces a stimulus, it must be an infectious agent. Yes, there could be the biological agents which trigger the injury to a cell or a tissue and then induce an immune response. But not all foreign intruders must be biological agents. You can also have physical injury. You can also have chemical injury, which induces an immune response. And for the immune system to respond, the innate arm and adaptive arm, there need to be specialized cells and also the chemicals which you talked about as, say, the plasma, the serum. So what I'm keen about is dragging your mind again back to that foreign intruder, which induces an immune response. So today we are going to understand if an immune response is induced. Now the body needs to protect yourself against that injurious agent. What do we understand with this term antigenicity, which is derived from an antigen, and immunogenicity, which is derived from an immunogen? So therefore, the immunogenicity and antigenicity. But before we go there, let's look at our learning outcome. At the end, you should be able to differentiate between antigenicity and immunogenicity, and you should be able to know the factors that influence immunogenicity, and also differentiate the properties of B and T cell epitopes. This I will elaborate on further what an epitope is, and even also expanding to you an explanation of what an immunogenicity and antigenicity is. But we need to understand some key things here which are quite critical to you. You must have understood, or you must have gone through the foreign intruder, as I mentioned to them. They are called antigens and they induce an immune system. So when you define an antigen, this is just a substance that can be recognized by the B cell receptor or by the T cell receptor. So we take note, the B cell receptors are those antibodies which are expressed on the surface of the B cells. But you must not fail to understand that some of the immunoglobulins or these antibodies secreted or formed by the plasma cells, of course, the mature B cells, they can be released into circulation and therefore recognize these antigens in circulation. So you should not be only thinking that when we talk about the B cell receptor, then there are antibodies which are expressed on the B cells. We have got also antibodies which are released into circulation, and they are able to recognize the antigens and neutralize the toxins produced from those antigens. We have got a critical thing for you to understand in the T cell receptor. That T cell receptor, we shall be understanding it deeply when we will be going through the third learning objective, differentiating between the epitopes. Epitopes, I don't want to start defining it now because it will not become clearer to you. We need to define it in the third slide also. And therefore, the T cell receptor recognizes this antigen, and they recognize only a portion of this antigen. So that portion of the antigen which is recognized is what is termed as an epitope. But T cells will not recognize these epitopes without a third molecule, and in this context, the major histocompatibility complex molecule. Who is teaching you major histocompatibility complex molecule? Is me teaching you a major histocompatibility complex molecules? By chance, I will, because it falls on 6th December when I will be on leave. But then you'll understand this is a critical molecule which is required for the T cell receptor to recognize the antigens. And therefore, we come to defining the immunogenicity versus antigenicity. The ability to induce this antibody or cell-mediated immune response, and cell-mediated immune response is associated purely with the T cells. The antibody-mediated immune response, also termed humoral immune response, is associated with the B cells. So the ability to induce humoral and cell-mediated immune response is what is termed as immunogenicity. Antigenicity is just ability of that antigen to combine with the T cell receptor or B cell receptor. Now let's look at this afresh because you are going to get mixed up. When we talk about immunogenicity, it's ability to induce an immune response. When we talk about antigenicity, it's ability to bind to the B cell or T cell receptor, the simplest definition. Immunogenicity, ability to induce an immune response. Now, this immune response can be humoral immune response or cell-mediated immune response. Antigenicity, ability of that antigen to bind to B cell receptor or T cell receptor. Stop there. So one thing you need to note, therefore, is all antigens would be able to bind to B cell receptor or T cell receptor, but they can fail to induce an immune response. Hence, all antigens can induce antigenicity, but they cannot be all immunogenic. All immunogens, those antigens which induce an immune response, fall under antigens. But only a small portion of those antigens are immunogens, which means they're able to induce an immune response. When we expand our terminologies in the context of understanding this immunogenicity and antigenicity, introduction of the term hapten, which is a small molecule that is in itself not antigenic, not immunogenic, which means it cannot be able to induce an immune response. But when we combine with a carrier molecule, this carrier molecule enables it to be now immunogenic. I repeat, haptens are usually used in the context of vaccine generation, and this is just that portion of the same microbe which scientists have identified is able to induce an can be conserved between different strains of the pathogen. Right now we talk about, say, the strains of the COVID virus in circulation, the Delta, whatever, Alpha, and scientists can look at the conserved region among these strains of the viruses. So it can be used to design a vaccine, which therefore can be able to protect you against the various variants of this virus. However, that conserved region is not able to induce an immune response, but it is the one which is suitable. Why? Because it is conserved and cuts across very many variants of this virus. So it can be the one to go for experimental study to produce a vaccine, but it is not in itself able to induce an immune response. So what it's done, it is conjugated with a carrier molecule. So this carrier molecule will enable it to be much more immunogenic. Hence, a hapten is a small molecule that is antigenic. Yes, it is foreign and is able to bind to B cell receptor, T cell receptor, but it's not by itself immunogenic. And if you combine it to a carrier molecule, then it becomes now immunogenic. Additional terminologies which I would introduce to you here, we've talked now antigenicity, ability just to interact with the antibody and or B cell receptor, T cell receptor, immunogenicity, ability to induce an immune response. And when we look now the link between haptens, immunogens, and antigens, we have told you that haptens are small molecules. They are antigens, yes, they're small molecules. In themselves, they cannot be able to induce an immune response. But if you combine them with a carrier molecule, they'll be able to induce an immune response and convert themselves, therefore, to immunogens. So these haptens and immunogens are antigens, but I've mentioned to you not all antigens could be immunogens. All immunogens are antigens, but not all antigens are immunogens. All antigens are not immunogens because some of them can fail to induce an immune response, but all immunogens are antigens. And I mentioned to you the aspect of the carrier molecule which you can conjugate with a hapten. So when, for instance, this portion of the hapten which you had identified again across variants strains or variants of a virus or say a microbe, and you want to use it to design a vaccine, and you conjugate it to a carrier molecule, supposing you challenge the whole component now to an animal model, it can be a rabbit or mouse model, you would find that in the body system of that animal model, there would be antibodies which are generated against now the hapten, antibodies which are generated to a carrier molecule, and antibodies which are generated against the conjugate, which means the hapten and the carrier molecule. And this way, we understand, therefore, that conjugating a hapten to a carrier molecule enhances the immunogenicity of that hapten and therefore can induce an immune response. Some of the haptens which have been developed and have reached, have been known to be utilized for induction of the immune response include DNMP, you have got protein carrier molecule or conjugate of it, and they form antibodies which can be against each of these components, like I've mentioned to you in this diagram, which is a cartoon diagram and simplified. So let's look at factors that influence immunogenicity. And I mentioned to you, I'm repeating here, immunogenicity is ability of a molecule or a substance to induce an immune response. What factors would influence such kind of immune response? One, how much that molecule is foreign from your body or the body of an individual who has come into contact with it. So far apart evolutionary lineage, the more immunogenicity that molecule will be. Secondly, we go to type of the molecule, and this relates to the chemical nature. You have got pathogens which constitute the body system, could be predominantly protein, polysaccharide, lipid, nucleic acids. And in that case, therefore, in terms of chemical nature, proteins are more immunogenic than polysaccharides, which are more immunogenic than lipids, and lipids are more immunogenic than nucleic acids. Molecular size. Larger molecules tend to be more immunogenic, and this can be illustrated with this cartoon diagram which I've shown you here. You conjugate the carrier molecule to Hapten to make it more larger, and in that way, it will stay much more or longer in the body system to stimulate the immune system. Again, composition, this relates to heterogeneity, which increases immunogenicity. You know the structure of the proteins, which runs from the primary structure, the sequences of amino acids, going to the secondary structure, the tertiary structure, and the quaternary structure, which is, when it reaches the tertiary to quaternary structure, it has got a lot of folding of the peptides which form that structure of the protein. Now, the quaternary structure is more immunogenic than tertiary, more immunogenic than secondary, more immunogenic than the primary structure, which is the amino acid sequences. Then degradability of that protein, how easy it can be degraded to be phagocytosed, and phagocytosis, I think, is ahead of, or you've not covered it, I would think so. So the easier this molecule can be degraded, the easier it is to induce an immune response. When we look at the physical form, the denatured form is much more immunogenic than native form. And this relates to the degradability, which we've talked about here. When the protein has been degraded to smaller peptides, it becomes much more immunogenic than when you consider it in the native form, which has not been degraded. So all these factors are factors which induce the immunogenicity, and more or less are used in understanding how exposure to pathogen or a molecule which is considered now an antigen can be able to induce an immune response. When we look at the concept of this, what we talked about in terms of the size of the molecule in the vaccine development, you know right now, progress has been made to come out from the whole organism to design vaccines which are more or less constituted through peptides. And when we talk about, say, the messenger RNA generated from the RNA viruses like the coronavirus to make vaccines, more or less we are at the level of the peptides, because you want to use a small unit of that composition of the organism to design a vaccine. So this kind of vaccine development concept has moved from the use of whole organism to toxoids to subunits to peptides. Very good. But in terms of immunogenicity, there is decreasing immunogenicity when you come from the whole organism. And this resonates very well to what I've explained to you above. The size of the molecule determines the immunogenicity of that molecule. The whole organism becomes much more immunogenic than the peptides. And hence there is need, therefore, in design of these vaccines, you would be having that peptide, but then also conjugate it to a carrier molecule. In this case, we'll understand now the scientific term when it comes to the design of the vaccine and the development. That carrier molecule is not any longer carrier molecule, but an adjuvant to a vaccine. Look at additional factors that influence the immune response or immunogenicity. We have got genetic factors. The way you'll respond to a vaccine, and for instance, you have now in clinics, people are talking about they had got different clinical symptoms when they got exposed to the COVID vaccine. So the way you respond to a vaccine is not the same way I will respond to it. This is related to the genetic composition of individuals. Again, you have got also a factor which is influencing immunogenicity, the dosage of the antigen, and this is why optimal dose is established to induce, to ensure that you tolerate it. The number of doses of the antigen which is being given, and this is not only relating now to the COVID vaccine, which is being given the second dose, the third dose, but you are quite aware of the booster vaccine, especially for polio and other viral vaccines which are in the market. The route of administration of the antigen also influences or determines the immunogenicity. You have got the intravenous way, which, and you definitely know when you are taught about the primary, secondary, tertiary. Yes, we reach tertiary. Primary lymphoid tissues, we get the secondary lymphoid tissues and the tertiary lymphoid tissues. I know right now you should be quite clear to understand that the mucosa-associated lymphoid tissues fall under tertiary lymphoid tissues. So when you inject the vaccine or when you administer a foreign antigen, it depends on where the antigen is going to be encountered faster by the immune cells, and especially the lymphocytes. So you have got the intravenous, then this would move very quickly to the spleen, and you know spleen as secondary lymphoid tissue. Then we have got subcutaneous, which still also have abundance of lymph nodes, intraperitoneal, oral, like the mucosa, and also inhale. The last two, you'll have a little abundance of the lymphoid cells. That's just why I mentioned to you that mucosa-associated lymphoid tissues have what? Yes, the lymphocytes, but not as abundant as you'd find them in lymph nodes and the spleen. So when you look at the route of administration of the antigen, then it comes down from the intravenous, subcutaneous, intraperitoneal, oral, and inhaled in that order. The point at which you will expose the antigen will influence the immunogenicity. And I mentioned to you again use of adjuvant in the vaccine development, a typical analogy to what we saw in the carrier molecule with the hapten conjugated together. But when now vaccine, real vaccine is being developed, we call it an adjuvant being added to that vaccine component and not a carrier molecule. And how does this adjuvant work? Adjuvants usually prolong the persistence of the antigen due to now the size of the molecule. Reflect back what we talked about, the hapten and the carrier molecule. So when you come here to talk about adjuvant, it's a replica of that carrier molecule, conjugate it with a vaccine. It will prolong the stay of the vaccine in the body system and give immune system more time to respond. It also increases the size of the antigen, definitely we've seen that, and this causes aggregation of the cell. And it stimulates the lymphocyte proliferation and activation. It stimulates local inflammatory response, resulting into formation of the granuloma, the activated macrophage cells, which move towards the site where stimuli has been recognized. It also enhances co-stimulatory molecules and predominantly the production of the cytokines to trigger movement of or to induce chemotaxis of cells to the site where the vaccine has been exposed or antigen has been induced. And I'm not going to go through this slide, but just to give you examples of some commonly used adjuvants in the vaccine development: alarm, incomplete fringe adjuvant, complete fringe adjuvant, bacterial lipopolysaccharides. This you can read at your own time. Why would one use an adjuvant, one adjuvant in preference to the other? Then I talked to you about the site of antibody or T cell binding to the antigen, and this is what I mentioned to you, that you have got only a portion of the antigen, or when you have got the B cell receptor or T cell receptor, it can only recognize a portion of this antigen. And that portion of the antigen which is recognized by the B cell receptor or T cell receptor is what is called an epitope. Or if you find epitope so difficult for you to pronunciate, you can use the term antigenic determinant. So an epitope is and same as antigenic determinant, and it is that portion of the antigen to which B cell receptor or the T cell receptor binds, or it is that portion of the antigen which is recognized by the B cell receptor or the T cell receptor. And it is the part of the antigen which will therefore determine the antigenicity of the antigen, and thus the term antigenic determinant relating to what I've given you explanation there. So one thing you need to know is that T and B cell recognize different epitopes on an antigen, and therefore we shall be looking at, remember I mentioned to you the first slide that I don't know, we'll get time to take you through the, I will get time to take you through the major histocompatibility complex molecules. So though I see the lecture is falling on time when I will be on leave, but you recall that I told you at initial slide that T cell receptor require additional molecule to recognize the antigen, and this is the major histocompatibility complex molecule. So we'll understand the differences between the B cell receptor and T cell receptor in terms of recognizing this epitope or antigenic determinant. Now, let's look at this, which is going to help us understand much more of this B cell receptor and T cell receptor recognizing an antigen or an epitope. We have got a virus, and I mentioned to you that the virus, you have got these spike proteins constituted with glycoproteins, and these are the regions which would be utilized by this virus to attach to the target cell, and it is purely composed with glycoproteins. So in these glycoproteins, there are numerous sites where it can be recognized by the B cell receptor and T cell receptor, which if I give you the last cartoon box there, you get several epitopes which are existent in these glycoproteins, which must be exposed so that the epitopes can be exposed and B cells or T cells can recognize. But that does not mean that this material in the nucleus of the virus cannot epitopes. It all which needs to be recognized. So to be recognized, this virus needs to be degraded so that the hidden in the genetic material will be exposed. But now I'll give you illustration with the glycoprotein, the spikes which are on the surface of the virus, which would be recognized by the B cell receptors and T cell receptors. And one thing, each different protein and glycoprotein of virus constitutes a different antigen or typically epitope. And each different antigen contains a number of different epitopes, which we are going to see now. In terms of recognition, how does B cell receptor and T cell receptor differ in what they recognize in this pathogen? Hence, the properties of B cell epitopes. One, they are usually dependent on the native. You remember when I was talking to you, a native versus denatured structure of the protein. So for the B cells, they will recognize that native tertiary, which means at the folding level, when the protein is still folded. So the pathogens will be expressing these proteins, but still at their folded structure or in native form. B cells will nonetheless be able to recognize the epitopes. However, these epitopes must be accessible, and usually they tend to be on the surface of the antigen, which means they are classified as hydrophilic, water-liking, water-soluble. They are more or less soluble. And they may be sequential, which means one antigen, then another, then they're joining together, or they may be non-sequential, in which case one antigen is found at a different point, another antigen found at a different point, and they're not joining together. So they can be made of sequential or non-sequential amino acid sequences. Epitopes which are made up of non-sequential amino acid sequences are usually also called conformational epitopes. So we have got sequential epitopes or conformational epitopes, and you'll see that in a cartoon diagram which I have ahead. But one thing you need to understand is that B cell receptors bind to soluble antigen, hydrophilic antigen, and there is no need of the major histocompatibility complex molecule as binary molecule. In T-cell receptor, we'll go into detail with that. What I need you to understand just right now is that for B-cell receptors to bind to their antigens, they don't need the major histocompatibility complex molecule. And large antigens will contain multiple overlapping B-cell epitopes, as we see in the subsequent diagram. So you have got this protein folding, and as it is complex in the tertiary structure or quaternary structure, B-cells will access its epitopes, antigens, and then bind to them. So there is no need of denaturation of this protein folded in its tertiary quaternary form for it to be for the B-cell receptors to recognize the epitopes. One thing you only need to know is, I think this will be learning again also later, antigen presentation and etc. These B-cells will produce numerous immunoglobulins on their surface, but these immunoglobulins which are attached on their surface immunoglobulins, are only specific to a particular epitope. If I look at this, I don't know which color is that, it looks red, no, pink. You got epitope there, pink, you got epitope dark green, and you got an epitope here, blue. Each of them has got key and lock compatibility to the antibodies which are produced on the surface. Hence, the second cartoon diagram is showing you the compatibility of these antigens which are in circulation towards the immunoglobulins which are expressed on the surface of the B cell, and they must be in sort of a key and lock model. And I mentioned to you that two kinds of B-cell epitopes exist. You can have discontinuous or conformational epitopes, or you have got the continuous or linear or sequential epitopes. And when I come to discontinuous, let's explain it first. You have got dotted epitopes which are spread all through and they're not joined all together. When you have got the continuous, which means the epitopes are lumped together and are joined together. This is the difference between continuous and discontinuous epitopes. But then you have also discontinuous, also called conformational. The continuous or linear is also called sequential epitopes. Now, you students, you are doing medicine, but sometimes you need also to think. So you don't need only to rely on what has been developed so that you apply. You have got, say, a test result which you have sent blood to immunology laboratory for diagnosis of, say, what can I give you? Let's even look at Salmonella typhi, or what other thing? Talk even any other thing. Say serological analysis of HIV virus. Now you have got 3 companies, and these 3 companies produce kits in the market which produce antibodies which are able to detect different portions of this pathogen which you are interested in. Now, one company produces a kit which has got monoclonal antibodies to detect for you an epitope 1. Epitope 1 constitute amino acids 1 to 12. Then you have got epitope 2 constitute amino acid 8 to 20. Epitope 3 constitute amino acid 19 to 32, 33. And the strain of these viruses which are in circulation in West Africa is different from the strain which is present in Kenya. Now you find out that, somebody has traveled from, say, Ghana. Ghana is in West Africa, and has fallen sick, and you suspect that maybe I want to test if Salmonella typhi, this person is sick with Salmonella typhi. But remember, the pathogen, the Salmonella typhi, assume the strain that is in West Africa is that which expresses epitope number 1. And you people in Kenya, the strain which is there expresses epitope number 2. So the kit in the laboratory which you have is that which detects epitope number 2, which this Ghanaian doesn't have. So you submit sample to immunology laboratory, and then the technical staff gives you result that, Daktari, this thing is positive. How will you make a judgment on a rational, correct diagnosis of this pathogen, noting that the epitope which you are detecting is that which spans from 8 to 12, yet you note the strain which is present in Ghana or West Africa is that one which is epitope 1, which is spreading 1 to 12. Take note that epitope number 2 overlaps. Amino acid 1 to 12 and then amino acid 8 to 20, it means 8 is already starting inside the epitope number 1. How will you interpret this result and start or institute management of your patient? And the report has come to you, and you as a doctor, you need to think beyond a technical staff or a technician. And you need to have knowledge of what you are treating. So here, a report has come to you, and you are being told it is positive. So you want to start treatment of this patient. But you have got some little knowledge that the strain of this pathogen, which is in your setup, is that which expresses epitope 2. And that which this patient could have presented with is epitope 1. In your present Kenya here, you have got kits which are detecting epitope 2. You take note that epitope 2 overlaps and starts at the inside of epitope 1. So tell me how you want to interpret this result and institute management of this patient. All right, we'll go on. Assume that you'll answer me in the card. When is the card scheduled? Let me see it. Sometimes you have a combined card, sometimes ahead. So I think that one forms part of what you will answer me, how you want to institute management of this patient with the results that you've been given from the laboratory. Then let's look at properties of T-cell epitopes. T-cell epitopes now involves tertiary complex and not a binary complex like we got the B-cell receptor and the antigen. Here, for the T-cell receptor, we have got a third molecule, major histocompatibility complex molecule. Nonetheless, these antigens or the epitopes must still be accessible, but they tend not to be on the surface of the antigen like we saw for the case of the B cells. Hence, they're termed to be hydrophobic, water-hating. So there may be internal linear peptides, but are found inside. You remember me when talking to you about the genetic material of the pathogen virus, you could have this pathogen, this epitope hidden in the genetic material of this virus, and the T cells need to recognize it. So the best way for the T cell to access this virus has to be denatured, and the epitopes which are hidden inside the genetic material be exposed. So the T cells will not recognize the antigens in their native form like we saw for the case of the B cells. So it does not bind to soluble antigen and requires processed antigen, which means denaturation. Hence, they will recognize only proteins, but some of the T-cell receptors can also recognize the lipids and glycolipids, and this must be presented on major histocompatibility complex molecules. Now, this is the tertiary structure. Just the tertiary structure of the T-cell receptor. You have got an antigen presenting cell, and this antigen presenting cell must express on its surface the major histocompatibility complex molecule. This you'll understand in detail. Why is it that when it is T helper cell, we need major histocompatibility complex class 2? If it were T cytotoxic cell, it will require class 1 major histocompatibility complex. But what I need you to understand today is this tertiary structure of the major histocompatibility complex class expressed on the surface of antigen presenting cell and anchoring the peptide, which now needs to be recognized by the T cell receptor. So you have a T cell receptor, it will not recognize this peptide unless this peptide is presented to it bound to major histocompatibility complex molecule. It can either be class 2 if it is presented to T helper cell, or class 1 if it is presented to T cytotoxic cells. Forget about this CD4 cell, which is a co-receptor. You will understand this when you'll be doing ontogenesis of T and B lymphocytes. So the clear understanding is, contrary to what we saw for the B lymphocyte, which is still also given here, the T lymphocytes will not recognize antigen in this simple manner, where you have got in circulation the antigens and are easily recognized by the surface receptor. The T cells must require the expression of major histocompatibility complex molecule for it to recognize that peptide. summary. One, we have to look at the definition of an epitope, which I've mentioned to you is antigenic determinant. And this is the part which the antigen is recognized with, or the recognition site on the antigen X, or antigen Y, or antigen P. And when we look at the B cells form bimolecular complex, and it binds to soluble antigen. There's no need of major histocompatibility complex involvement. The antigen can be protein, polysaccharide, or lipid, and the epitope must be accessible and often in conformational manner. And these antigens must be hydrophilic, which means motor liking. When we look at the T cell epitopes, we have them, T cell epitopes form a trimolecular complex. We've seen the T cell receptor, the peptide, and then the major histocompatibility complex molecule. So we have a trimolecular complex, and the T cells are able to bind soluble antigen, which means they are binding to hydrophobic molecule, and they require major histocompatibility complex. The antigen is usually a protein, but then the epitope, which is recognized, is internal. So the whole molecule must be denatured to expose that epitope to make it linear. And these epitopes are usually hydrophobic and must be bound to major histocompatibility complex molecule. To that end, I think I've taken you through the antigens, antigenicity, and immunogenicity, unless somebody has a question which I can answer. All right, if you don't have any question, I can release you for the day. You have a good day.
Thank you, sir.
Class rep, you'll remind me later for the notes.